All right, guys. Howdy. My apologies. I am still dealing with all the fun things related to the updates on the computers that are happening across campus. So I have to get the drivers installed for video through the office. So anyways, without further ado, I do just want to make sure that you guys have the last little bits of information for our chapter 7 so that we can complete chapter 8 on Monday and be ready to go for our exam next week. So with that being said, we had left off really focusing in on the specifics around utilizing invertebrates to determine the EOD, the earliest ovopositioning date, so that we can trace back to a potential PMI, right? And so again, I'm not going to go too far over this. I did edit out and edit down a lot of these slides for you. But just to reiterate some of the bigger concepts that we need to make sure we're aware of for the exam is we talked a little bit about how we do have odor mortis, and that odor mortis is specifically associated with the smell of death, and that there are several different factors that can relate to creating the smell of death. And again, most of this is through a lot of the breakdown of the body through the bacterial decomposition process internally, and also microbial and insects and everything else that's surrounding the body externally. So again, when we think about our VOCs, or we think about some of the concepts of what creates the off-putting smell of decomposition, again, questions I can ask you, what are factors internally and externally, right? We have to think about the soil content. We have to think about microbes within the soil. We have to think about microbes within the body itself. And then we also have to think about how the insects and the ovopositioning date, how these forensic entomological aspects add also their own secretions and their own processes to the decay process, adding and contributing to the smells of the VOCs. So some of the bigger ones that I definitely want you to be aware of are caverine and putrescine. These are sort of ones that we have already touched base upon in earlier chapters as well. If my thing will work, that would be great. And then we started to talk about how we could calculate potentially PMI from invertebrate developmental rates. And again, what we are now focusing in on, right? Chapter 6 was really about the different types of forensic entomological insects. Now we're focusing in on the distinct stages of insects and when they come to the body, and how we can use that. So again, the 4 major stages: egg, larva, pupal, and adult. And we must factor in that we are looking at not only these stages, but we are looking at temperature and environmental factors, because that is going to enhance, slow down, or confound how we develop what the earliest ovopositioning date is for the insects that are on the body. So the 2 major ones that we are concerned with for this chapter are flies and beetles. So again, maybe there is a classification chart like this. And so again, what are we looking at? We're looking at flies being our gold standard, specifically blowflies, because they are extremely well studied. And then we are also looking at beetles, because beetles are our late colonizers. They come to the body during the later stages of decay. So again, they provide sort of a nice overlapping timeline between fresh and dry putrid remains or skeletonized remains, right? So why, or again, we talked a little bit in class, you guys gave some great examples of how or what we could think about that could maybe affect the EOD. And again, it really comes back to temperature, environment, how the death occurred, what, or again, is the interface of the soil. How are the microbes in that soil affecting the interaction with the microbes on the body? And again, how is the insects that are feeding on the body factor into this as well? So the blowfly, we stated, is the gold standard. And again, it typically, what we know is flies are found in almost every environment on planet Earth. They can smell for several miles away. And again, they can come to the body and start laying eggs within 2 to 3 hours after death. But what we have also discussed as well in class is that wound myiasis, right? This idea that flies can actually start coming to wounds as a person may be moving towards death, or again, even in cases of neglect, that again may alter some of that. So we'll talk a little bit more about that as reiteration. So make sure that you're aware of the different stages of, again, the blowfly larva. Just what are some of the major characteristics of this? What is the overall life cycle? How do we define the pupal stage? And again, why this is important. The pupa stage is away from the body. Remember, after our third instar, we are moving away from the body. Again, also need to be aware of the generalized timeline of this, right? What is the generalized timeline as far as hours? And, you know, again, as we look at just the little asterisk, or not asterisk, the inference line here of about, right? So again, there's variation here, and that comes back to the variation between temperature and environment. So knowing what the pupa stage is, again, true or false, the pupal stage takes place on the body. It does not. Remember, after our third instar, we are moving away from the body to find dirt. We are finding soil to bury in very shallow, I should say, to be able to crystallize and go through metamorphosis. The tanning of the parparium is an indicator of age. From here, they move into the adult stage, and they again begin this cycle over. So again, the last little bits of this is once the adult fly emerges, typically our window is about 6 to 8 days. And once they remove themselves from the environment and leave, these pupal casings can remain for a very long time thereafter. So we had wrapped up sort of the summary and indicators of age. And we sort of then started to move into how to differentiate between PMI and time of death, right? Again, of course, we mentioned this all the way back in chapter 1, that these two don't necessarily always overlap or equate, because PMI is, again, the time that has elapsed since death. That is not when death has occurred. So again, when we are traveling or trying to find the EOD is helping us to potentially tell us a timeline of when or since when death has occurred. So again, when we want to consider this, we need to consider, do bugs have access? Do our insects have access? Were we in a shallow grave? Were we not? Do we have access via that? And then we need to go back to chapter 6. What are our types of beetles and or flies that can burrow? We know that for the case of our blowflies cannot do that, right? So again, if I was to give you some sort of case study that was discussing some aspects or in and around some aspects related to the blowfly PMI or blowfly EOD, and anywhere in that case study I had mentioned, buried in a box, buried under the soil, shallow, that would not, we would not be able to appropriately rely on that unless there was an indication that the body was moved from somewhere prior after death, to which blowflies basically hitched a ride, right? And were buried with the body itself or enclosed with the body, right? Those are really the only times that we would ever see that take place. I sort of edited this one down, and I said that really, while I won't ask you to participate in creating this or being able to appropriately identify this, this could just be simply a question of which of the following is a potential method right, for calculating times since ovio positioning. So again, the isomelogram diagram method, this is just a plotting of temperature, time of hatchlings, the overall temp in Fahrenheit versus Celsius, and again, what is the potential timeline of days of hatching? It gives us an XY indicator of which we can then form a regression line. So things that can, this was the last little bit of information that we covered when we last met. Again, complicating factors that can result in the underestimation of EOD. So I am delayed in actually what the EOD timeline is telling me. So again, I had just mentioned it, fly access. We didn't have access to the body. Season is too cold. Body may be stored in some sort of compartment or is in a freezer, right? If the body is then moved and is able to come back up to temperature and have accessibility, right? That is going to give a delay for potentially what PMI is relating to EOD. Insecticide sprayed on the body may keep our insects away, which again creates that delay. Larva or pupal diapause, dormancy related to environmental conditions, again, too cold, too violent of weather. And again, this is such a bummer that for Wednesday's class, you guys didn't get a chance to go out because, right, you know, it would have been great to actually be able to see that right before a giant rainstorm hit or in between the timeline of the weather that we've had to see how it did disrupt the insects that were feeding on it. Because again, heavy rainstorms, flies are not going to fly, right? Or maybe flooding occurs such that it washes away larvae. And so I'm really bummed that we didn't get a chance to do that, but we'll go out on Monday and we'll see what we can kind of come up with. And finally, again, predators. They may alter the developmental appearance for those that are remaining. Factors that result in overestimation. I'm too far ahead in my timeline, actually, and I need to be further back. So again, wound meiosis. Flies were able to be attracted to the smell of rotting flesh. Prior to death, flies began to feed and lay eggs. And again, maggot activity proceeded before death. Development of the maggot mass feeding can raise body temperatures, can give a false developmental progress report. And again, minimum temp of development, different regions, different species, things can travel from one to another, biological swing, all of these things need to be considered. Things that can complicate either over or under, drugs and toxins, and of course the microclimate of the body. So what I did for you is I just threw this in, hint, hint, nudge, nudge, wink, wink. This would be a great thing for you to practice just filling out on your own, right? Don't have your notes open, don't have this PowerPoint or video open, but maybe you want to print this out and maybe then you want to utilize coming down the pipeline, right? This as a study tool. This is just a follow-up that we have talked about some of these, right? Really the ones that were presented would be the big ones I would be pertaining to asking you as it relates to questions on the exam and or that Venn diagram. So I apologize, I don't know why this came out this way, but again, this is kind of the movement or how we can show that, you know, we can see that insects can affect the output of what we are finding on bodies. So we can dress pigs up in different types of clothing. And again, this can alter the appearance or accessibility. Additionally, what we can also find is that if we allow this, right, insects are still going to be attracted to this. So if that is the case, then what we will see sometimes in cases of things like sexual assault, where maybe we have misidentified. There was actually a case where forensically investigators, or I should say researchers, dressed pigs up in pig cadavers up in, you know, miniature sort of doll like or girl and boy young adolescent young children clothing wear. And what they were seeing was, does the invertebrate activity right? Because eventually it becomes such a feeding mass. Can the movement of the larva or insects on the body give a false indication that maybe sexual assault took place, right? Pants pulled down, skirt pulled up, underwear moved. And in fact, they did find that insect activity can give false appearance of this. Now, that's of course going to be extremely important if we're looking at the cases of finding this within the early stages of decomposition. But we do have to consider this. This is something we need to keep in mind. So, summary of insect evidence that suggests that a body was moved after death. These are most of the things we have discovered or most of the things we have discussed. And again, this is just an overall summary from the book. So how do we then utilize this information and knowing that, again, as insects are feeding, that the invertebrates themselves can actually be consuming the drugs, toxins, and or other chemicals within the body itself, right? So if the body is in later stages of decay, maggots and their puparia cases should be tested. Concentrations found between individuals and invertebrates at this moment is not universal. It's just an indicator, right? So taking things like the entomological study of these things, looking at the toxicology report, looking at environmental factors, we can come up with entomoxotoxicology, which is looking at how do we apply the take-up of drugs and external environmental factors, right? Natural occurring, xenobiotics, things of that nature, that could influence the rate of development, but also could be a holding tank as an indicator of exposure while alive or what led to death. So again, it's not an absolute, it is simply just an indicator. We also need to extract DNA from all maggot casings or beetle crops, as this may help identify when there may not be a body present, but there are many insects that are still found within a given area. Where potentially there is a suggestion that insects crawled into a body from elsewhere, meaning again, insects that are accidentally associated with dead bodies, or that insect samples may have been accidentally switched once that forensic evidence has been collected. Sometimes human error occurs, right? So again, this could be a great type of question. Walk me through why DNA extraction from invertebrates, from maggot or beetle crops can be helpful in certain types of cases. Can you give me 3 examples, right? These would be those 3 examples. Additionally, things like fleas and lice can interact or indicate, I should say, the transference between individuals through blood meal sharing, right? Pubic lice can and sometimes has been used, again, with a collection of lots of other evidence that we can showcase, we can indicate that individuals had close contact with each other, right? Again, this can be transfer between assailant and victim, and it may carry blood meal from the assailant and also from the victim that could then link those two together. This is all supporting, it is not necessarily going to be the end all be all evidence, right? Lots more research needs to be done here. Sorry, I don't know why that's taking so long. Okay, so the last thing, again, in terms of that is where are we getting this information from? So inside of our maggot casing or inside of our maggot, we can actually see once they have consumed with inside of the crop there, it's kind of an easy indicator to look at the maggot itself and see that they are holding onto a meal with inside of the crop itself because it's very surface level. Right below sort of the epithelium of the maggot. They're not, they're soft bodied, they're sort of in a translucent type of appearance. And so we can remove their food source, right, that's now being stored and actually test that for human DNA. All right, the last little bits of this chapter relate to how do we appropriately collect invertebrate evidence for forensic investigation. So the most important part here is we are referring to what is called the chain of custody. This is now the record keeping and of all the different phases of collection, whether that's on site with the little cards that you see with the numbers, to the photographic evidence, to the retaining of individual bags, to each piece of evidence. It needs to be categorized, marked, and it needs to be then recorded. And everybody that is a part of the investigation will have a specific format for their area as part of the chain of custody. Representative samples of all invertebrates found on, in, and around the body will be collected and put into separate containers, right? That would make sense. We would want to retain non-cross-species interaction, predatory behavior, anything of that nature, more importantly, cross-contamination of any sort. All specimen collection needs to be kept separate. Maggots found in the mouth will need to be separate from maggots found in and around the gut, right? That would make sense because, again, microclimate and developmental stages may be different. Also, stages of decomposition may be different. Ideally, maggots should be killed on site, some, right? And that time specifically noted for preservation purposes. If later, the time, location, and temp should be noted, because again, we may not get to the body right away. We also would want to do site kill, and we would probably also take back subsamples to rear to maturity to verify species and to also help in indicating timeline, developmental timeline. So once back at the lab, we would, sometimes the sort of accepted method in the field is to rear or grow from eggs to adult, kept in a controlled group to base, should be kept into a controlled environment, and there should be a control group to base that timeline against, right? So there's going to be already growing maggots or larvae, eggs to adulthood in a control incubator that will then begin to be utilized at the same timeline that we are bringing in those from the site itself. Typically, they're going to use a very simple, economically friendly methodology, which is plant propagators. This will keep a constant temp and condition. And obviously, we have to ensure that nothing else can get into this, no other flies, because, right, they're going to be attracted to things that we're going to use to rear, right? Insects then are transferred into individual tubes during the rearing process. They are then killed immediately and recorded for environmental time and conditions, and again, based off of what they found at the site itself. So for preservation, if things are hard body, they are typically going to be pinned for identification. Small non-hard bodied invertebrates, these are going to be stored, or I should say small hard-bodied invertebrates are going to be stored in a 70% ethanol solution. And it will be somewhat similar for those that are just soft-bodied as well. So again, soft-bodied invertebrates, maybe near boiling water, but we would preserve within acetic acid or acetic alcohol or an alcohol solution to keep sterile. With our hard body invertebrates, again, there's lots of different methodologies we can use, but again, for preservation, we can either pin them, still use acetic alcohol, or a 70% alcohol. So this is just a follow-up of sort of the succession wave. And this video talks a little bit about the forensic entomology at crime scenes. So that is it. That is everything that you would need For this next exam, and I think I also have one more in there. Forensic entomology experience at the North Carolina Museum of Natural Sciences. Just another reiteration of how we kind of use these things and store and maintain them. Just fun stuff for you guys to look through. Overall, though, that is it. That's everything you will need. And from here, I will post this and hopefully you have time to review that. We will finish with vertebrates on Monday. We will also check our pigs and we will be ready to hit the ground running for our exam 3 on Wednesday. All right, guys, have a great weekend and I will see you next week.